Welcome to biochemistry, your next stage in your chemistry career. You've learned about organic carbon-based chemical reactions. In this quarter, you're going to learn about the carbon-based reactions that occur in organisms, that occur in you. In particular, in this quarter, you're going to learn how those chemical reactions, how those rates of those chemical reactions are controlled. The rates of those chemical reactions and the chemistry that occurs is called your metabolism. The chemistry that occurs in your body is simple yet extremely complicated. It's simple in regards to the type of chemical reactions that occur. The number of different types of chemical reactions is relatively small. You learned in your organic chemistry class most of the chemical reactions that happen in an organism. What makes it complicated is how those chemical reactions are linked to each other. If you look at the poster outside our laboratory, when you get a chance, there's more than 1,000 reactions on those posters. Each arrow represents a chemical reaction, but those chemical reactions are linked. When you took general chemistry and even a little bit in organic chemistry, you've thought of one reactant, one product. It's a little different than that. Most of the products that are formed in your body become reactants for another chemical reaction. And we'll learn about pathways in just a little bit. A strategy for the chemical reactions in your body is to keep concentrations constant. Most chemicals in your body that aren't waste molecules, those concentrations are relatively constant. To keep a concentration constant for any particular molecule that's in your body, the strategy for an organism is to change the rates of chemical reactions. Meaning, if you had a molecule, your body has the ability to control the rate of the chemical reactions that produce that molecule, and the chemical reactions, you have the ability to control the rates of chemical reactions that use up that molecule. Back on the title page, I'm going to give an analogy of a lake and rivers flowing in and out. Think of a concentration of a molecule like the lake level, like Lake Coeur d'Alene. So say we had a lake, and this could be like Lake Coeur d'Alene, and it's at a certain level. There's water flowing out, the Spokane River. There's a dam at the head of Lake Coeur d'Alene that produces the Spokane River. So this is like our river out. There's water flowing into the lake. So this is our river in, like the South Fork of the Coeur d'Alene River. And there's rain that falls in there. Avista tries the best they can to keep this lake level constant. So this lake level here, this is like the concentration of a molecule. So the river in, this is analogous to chemical reactions that are producing some sort of molecule. The river out are chemical reactions that are using up that particular chemical. But let's just say the lake level was constant. So the water flowing in and the water flowing out was the same. So you have the same level. There's a dam on the Spokane River for the water flowing from Lake Coeur d'Alene to produce this Spokane River. What if all of a sudden that dam was opened wide up and the river out that increased? But the river in, the water flowing in, was constant. What would happen to that lake level? And most of you could figure that out. The lake level would decrease. That lake level decreasing is like a concentration decreasing for a molecule in your body. And a molecule we could think of in your body, maybe glucose. And we'll give some other examples of it in just a second. What if the river out, what if it was constant? And let's say we're at a certain lake level, but there was a constant flow out. What if we had a gigantic rainstorm and the water flowing into that lake level increased, but that dam, it remained in the same position, so the water flowing out was constant? What would happen to the lake level if all of a sudden you have a big increase of water flowing in? Its level would increase, the concentration, if this was happening to a molecule in your body, the concentration would increase. So let's think of some examples in your body, and we'll see this chemistry later on. We're just right now trying to get some sort of basic understanding of how concentrations are controlled. Well, glucose, there's blood glucose. And what's amazing, if you remember from your 121 class, if you took it from me, there's about 4 grams of glucose in your blood. There's about 20 grams of glucose from starch in a piece of bread. So there's not a lot of glucose in your blood. You can get more blood glucose from your diet. That's like a river in. You also have the ability to synthesize glucose in your liver. And the liver can secrete glucose into your blood. You'll learn more about that later. Blood glucose can go into a lot of different cells. So it can flow from the blood into different cells. That's like using it up. What if all of a sudden you go home today and you have a gigantic candy bar and there's 1,000 calories worth of glucose in it? All of a sudden you increase the glucose into your blood. If this dam that was controlling the glucose going into your cells, if that remained constant, what would happen to the blood glucose? This is analogous to having that torrential rainstorm and a lot of water flowing in. That blood glucose concentration would increase dramatically. What happens when you increase the blood glucose? Somehow that blood glucose interacts with the cells, and the transporters of glucose into the cells, they work better, and it's like this dam opens up and the blood glucose can flow into cells. Your body is amazing. If a concentration changes of some molecule, the pathways that produce or use up that particular molecule are regulated. They either increase or decrease to try and keep a concentration constant. We could kind of see an opposite effect if blood glucose concentration decreased. Let's say you started to exercise and the usage of glucose in your cells increased and you started taking more of the blood glucose from this lake. Let's say you weren't eating, so maybe your from your diet, you weren't bringing any glucose into your body while you were exercising. What happens, and we'll learn how it happens later because it's pretty complicated, but your liver would start producing more glucose and secreting it into the blood to try and keep that lake level constant. This is like, what if we were using more water in the Spokane River? What if we could tell nature to start raining more to produce more water into the Lake Coeur d'Alene to try and keep the lake level constant? It's pretty amazing, and by the time we're done with this quarter, we'll see how that regulation occurs. And it's not just one step, it's pretty complicated. But as far as analogies, we have our river in, we're river out, and we have our lake level. So think of the lake level as analogous to the concentration of a molecule. The river in, that's the production. So how much of that molecule is being produced? The river out is usage of that molecule. So river out, usage go hand in hand. If there's a dam, and the dam that controls the river in, there could be dam control points on, and we'll kind of go over to there, on the river in too. And also this analogy up here, we're showing one river in, one river out. There could be multiple, and there usually is, multiple river ins and multiple river outs. When you get a chance to look at that metabolic poster outside the hallway, outside our laboratory, and if you went and looked for glucose or some other molecules, like we'll see in this chapter, like acetyl coenzyme A, you'll see a molecule, and there could be lots of arrows pointing away from it and going towards it. Each arrow on there is a chemical reaction, but a molecule can be used for a lot of different pathways. It gets pretty complicated. One molecule isn't used just for one thing. But as we go on through this course, having this analogy and understanding it is pretty important. And we'll use it a lot when we're describing chemical reactions and control in your body. So to summarize some of the bullets we just said on the previous page, metabolic pathways, they're regulated. And we're going to see how they're regulated. It's kind of complicated yet not. And it has to do with proteins, which we're going to learn about in the upcoming chapter. So it's hard to say at this point or to describe how they're regulated since we don't know enough about proteins to understand why. But we'll see that and we'll talk more about that as we go on through the course. There's quite a few different types of reactions. So the number of reactions there's thousands of different reactions. But as I mentioned earlier when we started, the different kinds of reactions, what functional groups are reacting, that's relatively small. So the mechanisms, the types of chemical reactions is pretty simple. And there's some very, very common molecules, and we'll see some of those later on in this chapter, that we see in all life forms. The type of chemical reactions that happen in your body, you're pretty similar to a bacteria. I mean, you're a little more complicated. Bacteria is a single cell organism. You're a multi-cell with different types of cells. But the types of chemical reactions in that cell, they're very, very similar. What makes you complicated is the chemical communication that happens between your different cells. Like your thinking making your muscles move. Well, a bacteria doesn't have different cells like that. So you have chemical messengers that we'll learn about later called hormones, neurotransmitters. There's a variety of different types. So there's thousands of different types of reactions. What's also interesting is virtually all these reactions have enzymes. And we'll have a chapter just on enzymes. But in your body, the vast majority of these enzymes are proteins. And if you remember from your general chemistry, enzymes, these are catalysts. And they help increase the reaction rate. They can increase the reaction rate by about 10 million times. What's interesting for some of the enzymes that are in your body, some of these are the dams. How well they work can be regulated, can be controlled. And again, we need to know more about proteins to understand how enzymes, which are proteins, are regulated. Something else that is also true about all cells for all organisms is that you take in, and all cells do, take in fuel molecules, and the chemical energy from those fuel molecules is harnessed to do reactions that are in your body. This is true for all organisms. All organisms have the ability to metabolize glucose. It doesn't matter what type of cell you are. Any cell that I consider alive has the ability to take glucose and take chemical energy from glucose and do the type of regulation and production of molecules to produce life. If a cell didn't have an input of chemical energy from fuel molecules, the cell wouldn't survive. If you remember from general chemistry, what is chemical energy? Remember, chemical energy is the potential energy from electrons. Electrons are what contain the potential energy, the chemical energy in a molecule. And I'm not sure how much you remember this from general chemistry, but if you had a proton, or you had protons in a nucleus and you had an electron, there's an attraction between the two. Anytime you have attraction, you have potential energy. This is no different than you being on the second floor in our science building versus the third floor. You have more potential energy when you're on the third floor. If an electron moves closer to the nucleus, it releases potential energy. That would be the same thing as you going from the third floor to the second floor. The point is, and for this class, taking electrons from one chemical to another, that's taking the chemical energy from one molecule. When glucose goes into your cell, it gets oxidized. Its electrons are taken, and that chemical energy is used to do other chemical reactions. And we'll talk about that a lot in this quarter and in this chapter. Shown on this page is an example pathway. And the name of this pathway, a lot of pathways have names. The name of this pathway is called glycolysis. This is a pathway that occurs in every cell in your body. In fact, it occurs in every organism, everything that I consider an organism. To start this pathway is glucose. Well, it's a starting molecule for this pathway. There's a lot of acronyms on here that I don't expect you to know. This is just an example pathway. Glucose, when it first enters a cell, it reacts with a molecule called ATP. And we're going to see the structure of ATP in a little bit. This acronym stands for adenosine triphosphate. This is the very first reaction that happens to glucose in any cell in your body or in any organism. It makes glucose 6-phosphate. A phosphate is transferred from ATP to glucose, and you form a phosphate ester. A carbon-based molecule is produced. In a pathway a carbon-based reactant that's produced, well, it's a product in reaction number 1. In reaction number 2, it becomes a reactant. The vast majority of all the molecules in your body that are produced, that are products in some reaction, they're a reactant in another reaction. So glucose 6-phosphate's produced, but then it's used up in reaction number 2. What's produced in reaction number 2 is fructose 6-phosphate. It's the reactant in reaction number 3. So this is a common theme for pathways. Some carbon-based molecule is produced in one reaction, it's used up in another one. In reaction number 3, this fructose 6-phosphate gets another phosphate attached and it forms fructose 1,6-biphosphate or bisphosphate. This fructose 1,6 compound is a product in reaction number 3. It's a reactant in reaction number 4. And we can keep going and going, but this is considered a pathway. The final molecule, the final carbon-based molecule that's produced is called pyruvate. It's a reactant in a few other pathways, and we'll see those later on. But this is the end part of this pathway. When pyruvate reacts further, it's considered to be a reactant in another pathway. The only molecules that are produced in some pathway that aren't reactants in another pathway are your waste molecules. And a good example is carbon dioxide. When you completely break down a fuel molecule, all those carbon atoms become carbon dioxide. It's a waste molecule. It's not a reactant in a pathway. So you're going to see carbon dioxide produced, but you're not going to see it as a reactant in a pathway. So you're not going to see an arrow coming from it. Shown down here is something that chemists call the net reaction. So in a net reaction, it doesn't show all the molecules that are produced then used up. So since glucose 6-phosphate is produced, but it's used up, you don't see it down here in the net reaction. Same thing for fructose 6-phosphate. It's produced, but then it's used up, so you don't see it down here. It's kind of like a math equation. If you had these 10 reactions, and you're going to get the net reaction, if something's produced but then used up, it gets marked off and you don't see it on the net reaction. Pyruvate, for our pathway, we only see it being produced, so you see pyruvate down here. Glucose, you see it up here being used up, you don't see it being produced, so you see it down here. There's also some further vocabulary that I need to tell you about and explain because it's going to be presented this way a lot. So on this page, we see chemical reactions. Hopefully it's what you're used to. Well, in biochemistry, we add to it. And what do I mean by that? Well, when you write a chemical reaction, say we're starting with reaction number 1, we have glucose and it's forming glucose 6-phosphate. So we'll write all that out. And make sure that's an A. When you write chemical reactions in biochemistry, often it's presented in a different way. The carbon-based molecules that are produced then used up are written in the normal way with that arrow. The molecules that are reactants and products, biochemists usually add additional arrows. And you may have some sort of looped arrow looking like this. The arrows always point towards the products. So this arrow pointing right there, that's producing to the products. There's 3 products in reaction number 1, glucose 6-phosphate, ADP, and that H+. It's glucose 6-phosphate that's a product there, and it's going to be the reactant in the next one, and it's the one that goes on the main arrow. The other 2 products, ADP and hydrogen ion, they're also products, but they're put as part of that looped arrow. The carbon-based molecules that are produced, then used up, stay with the solid straight line arrow. For reaction number 1, glucose and ATP are both reactants. It's glucose that's the carbon-based molecule that goes on the main arrow. So if this was reaction 1, reaction number 2, well, there's only one reactant, one product, and both of them would be on the main arrow, so fructose 6-phosphate. So this was reaction number 2. Fructose 6-phosphate's produced, but it's a reactant in the next one, so it's on the main arrow. For reaction number 3, fructose 1,6-bisphosphate is the product, and it's the one that's going to be the reactant for reaction number 4. So it's the one that would go here. And this is reaction number 3. There's also other products and other reactants, so we can make our looped arrow. Okay, the fructose 6-phosphate for reaction number 3, it reacts with ATP. So there's 2 reactants there. There's 3 products. There's also ADP and a hydrogen ion. But it's the fructose 1,6-bisphosphate that's the reactant for the next one, and it's the one that goes along the main arrow. The point is, this format, instead of having 10 separate reactions like we're showing here, it's a way to link all the reactions together. The carbon-based molecule, again, that's a product, that's a reactant in the next reaction, is the one that goes along the main arrow. The other products and reactants that are not used in the next reaction, they go on the looped arrow. And we're going to see that format a lot this quarter. So here's an example of a pathway, and we're going to see this pathway in some upcoming chapters in upcoming exams. What I've shown you on this page, I don't expect you to know the details of this glycolysis pathway. Note, there's a lot of practice problems that are given in your study guide, example practice problems. Remember, there's a key posted on Canvas. Use the practice problems to guide you towards the type of questions that'll be on the exam. There's always a lot of information that I provide in the lecture notes, and trying to guess what you're going to be tested on, that could be quite difficult. Use the practice problems to guide you, to allow you to understand what you're going to be tested on. They're extremely helpful, and I'll remind you in lecture, and I'll remind you in the laboratory. In this table, there's also the enzymes. This is nothing you need to memorize, but here are the listing of enzymes. In fact, this table is in your information packet. Not exactly the same thing. There's something called inhibitor and activators in this table. There's some memorization in this class, but it's still very analytical. And just know what's in your information packet to make you know what you need to memorize and not to memorize. What's also listed in the table that's in your lecture notes is the free energy change. This is the energy change for the reaction, and we're going to talk more about this in some upcoming slides in this chapter, which is something I do expect you to know. You don't have to have these numbers memorized, but just remember the negative and the positive energy changes. This is the energy that's either released to the surroundings or taken in. In your body, in your cell, your surroundings is the water molecules, your cellular fluids. This quarter, you're going to learn about a lot of different types of pathways. Eventually, you're going to make a poster and put all those pathways together and link all the chemical reactions. When you have pathways, there's 3 main types that look a little bit different. And these 3 main types, one of them is called a linear pathway, another one's a cyclic pathway, and another one is called a spiral pathway. We'll see all 3 this quarter. The differences is when you go through the pathway, the reactants that become products, the products become reactants. They're slightly different. When you have a linear sequence, and a linear sequence is just like the one we saw on the previous page for glycolysis, but you have some starting molecule, some A molecule, it forms a B molecule, and that B molecule that's a product becomes a reactant for reaction number 2. There's enzymes for every reaction, and they're different enzymes. If you looked on that metabolic poster in the hallway, you would see arrows, and on top of it, on top of each arrow is the name of the enzyme in blue. There's a different enzyme, it's a different protein for every chemical reaction. So enzyme 1 is for reaction number 1, enzyme number 2 is for reaction number 2. And then that C that's produced in reaction number 2 becomes a product. And eventually we get an end molecule that could be a waste molecule if it's something like carbon dioxide, or it could be a reactant in another pathway. But there's an example of a linear pathway. So when you look on the poster in the hallway, and which will go on your poster at the end of the quarter, and when you just have one straight line and forming something else, that's a linear pathway. A cyclic pathway, so if you look on that poster in the hallway, you see some circle, and there's a few. It's called a cyclic pathway. And let's give an example of how that works. So we have some sort of A reactant starting it, and it reacts to form B, which is kind of like a linear one. B becomes, it's produced, it becomes a reactant for reaction number 3, and we form C. Just some A, B, and C, just some general chemicals, could be anything. These C molecules react and produce D. What's interesting about a cyclic pathway is those D molecules reform A, and you can go through the pathway again. That's called a cyclic pathway. If you took general chemistry for me, we had that one lab where we had a cyclic pathway forming different colors, and we reformed the starting color. That's a cyclic pathway. If you looked on the poster in the hallway and looked up the name, because the names of the pathways are also given, it's called the Krebs cycle. That's one we're going to be seeing. We're going to also see the urea cycle. They're all cyclic pathways as we go through this course. A spiral pathway is kind of like a linear one, but it's just a little bit different. And if we look at it, let's say we started with an A molecule, and it reacts kind of like in a linear one and form B. B becomes a reactant and forms C. And then we form some other molecules like D. What's interesting about this one is these D molecules, and there's also going to be some E molecules. One of those that's produced here goes back through the whole pathway again. And a good example that we'll see this quarter, we're going to look at the breakdown of fatty acids, eventually into carbon dioxide. But when a fatty acid, it's a carboxylic acid that could be around 20 carbons, 16 carbons. But every time that fatty acid goes through the pathway, 2 carbons are cleaved off. So if you started with 16 carbons, so 16C, and if it went through, the 16 carbon fatty acid would go through. 2 of the carbons get cleaved off and go somewhere else. If you cleave off 2, there's 14 carbons. Those 14 carbons can then go back through. Every time it goes through, 2 carbons are cleaved off. Eventually, you're left with just 2 carbon segments. But that's a spiral sequence. So it's a little different than a cyclic. You don't keep going and going and going. When you go through a spiral one, eventually, if you're breaking it down, you're cleaving them apart. We'll also see the opposite. Your body has the ability to synthesize fatty acids. It's a spiral pathway. It's just the opposite of breaking it down. Every time the molecules go through, 2 carbons get added on. But that's called a spiral pathway. You use the same enzymes every time you go through. Another general thing about pathways in general, some are called catabolic and some are called anabolic. When you have a catabolic, for that particular pathway, that's large molecules being broken down into smaller molecules. Whenever you have a catabolic pathway, energy is released. And that energy that's being released, some of it could be just heat to warm up your cellular fluids, or more importantly, the chemical energy that was in the larger molecules gets transferred to some other molecule. So larger molecules forming smaller molecules is called a catabolic pathway, and that plus energy means that chemical energy is being given to other molecules. And just to give you an example, it could be something like glucose with those 6 carbons, and we're just kind of given the net process forming carbon dioxide, so there'd be 6 carbon dioxide and other things. But you'd also have some heat, some energy warming up your cellular fluids, plus chemical energy that was given to some other molecule. So that's a catabolic general direction. We'll put a circle around that. Anabolic would be smaller molecules making larger molecules. And a good example that we'll be learning about in the next chapter are amino acids. And you may not know what amino acid is yet, but that's okay. Amino acids are used to make proteins. The proteins are big polymers made from amino acids. When you go that direction, you're going from smaller molecules to larger molecules. When you're going that direction, you need energy. So doing the reverse process. And energy, how does your body get the energy to do this? Well, you don't have Bunsen burners heating up your cellular fluids. To do this, you need chemical energy from other molecules. Your body doesn't use the kinetic energy of your cellular fluids to make bigger molecules. If that's the case, that would make your cellular fluids get really cold, taking the kinetic energy from those cellular fluids like water. Whenever you're building bigger molecules, it requires energy, but it requires chemical energy. So the transfer of chemical energy, potential energy from some other molecule to make the bigger molecules. And you can think of it analogous to a spring, a spring that's cocked, that contains potential energy. When those electrons go from one molecule to another, it's taking the potential energies of those electrons. It's like taking the potential energy from a spring to do some sort of work. Chemical energy, taking it, is taking electrons. And we'll see those type of reactions as we go through our various pathways. Shown on this slide is an example of a cell. And this isn't biology. I don't expect you to be able to draw a cell and explain every part of the cell. But you do need to know some of the vocabulary as we present information. So as always, do your practice problems to guide you on what you need to know. But we're going to look at eukaryotic cells, so the type of cells that are in your body. And there's some differences if you're a plant cell, but don't worry about that. But everything that I consider alive is a cell, and it has a membrane. And this membrane, which we're going to learn about in much more detail later on in the lipid chapter, it is the portion of the cell that allows molecules to go in and out. It's very selective with the type of molecules that can be transported in or transported out. We'll learn in a lot more detail about this transport process, but for this chapter, for exam 1, just know that all cells have cell membranes, and it's this portion that's selective on the molecules that can make it into the cell and can make it out of the cell. Another thing about the cell, the liquid portion of it is called the cytosol. Okay, so what I wrote down here, it's the cytosol is the liquid found inside cells. It's usually H2O. Usually H2O is a solvent, and that just means that's the component that's highest in concentration in that cell. Sometimes it's not that way. Most of the time it is. And there are thousands of different types of solutes. So those are other chemicals that are dissolved within that cellular fluid. When we eventually start talking about pathways more specifically in upcoming chapters, some of the enzymes in certain pathways are in the cytosol. And some are found within other structures like the mitochondria. And we'll talk more about the mitochondria and describe it in upcoming chapters, but it's considered an organelle. An organelle is a structure within the cell, and these organelles also have membranes, and they serve different functions. There's some enzymes for certain pathways that are found within the organelle. And the only organelle we're going to, for the most part, talk about in here is the mitochondria. There's other things, some of these other structures that, the chemistry is very important, but we just don't have time to get into that chemistry. That one cyclic pathway that I mentioned earlier called the Krebs cycle, those enzymes are found within the mitochondria. So when we talk about the Krebs cycle, we'll see more about the mitochondria. The cytosol, the enzymes for glycolysis, are found in the cytosol. You don't need to know that yet. You will a little bit later. And what I wrote down here, the mitochondria is an organelle, and organelles have membranes. The composition of the membrane in an organelle is different than the cellular membrane. And what can go in and out of a mitochondria membrane is different than the cell membrane that it's part of. Lastly, there's something called the cytoplasm. And let me read what I just wrote. The cytoplasm is everything inside the cell except for the nucleus. So here's our nucleus right there. The nucleus is what contains your DNA. And we'll talk a little bit more about the nucleus when we get into the protein chapter. The proteins are synthesized based upon the structure of your DNA. So cytoplasm would be the cellular fluid and all of the organelles. And don't worry about some of the other things that are outside the cell. We won't be talking about that in here. This quarter, when we discuss metabolism, you're going to learn a lot of fundamental aspects of how the chemistry in your body is controlled. we could pick any pathway or pathways to describe this. For this class, I'm picking fuel molecules. So we're going to look at catabolism and anabolic pathways for fuel molecules. So what are fuel molecules? Well, those are proteins, carbohydrates, and something called triglycerides, which are fat and oil molecules. When we look at carbohydrates, there's a lot of different carbohydrates, and you learned about that in Chemistry 122. But the starting molecules we're going to look at and the ones you're going to put on your poster, well, one of them is glucose. Another one is fructose. And the big polymer we're going to look at is glycogen. Triglycerides, and you may have a hard time reading my handwriting, but triglycerides. Triglycerides, those are fat and oil molecules. And we'll see the structure of triglycerides in the lipid chapter. In our poster, when we're done, we're going to look at the catabolism, the breakdown of triglycerides, and the formation of triglycerides. Proteins are considered fuel molecules, but in your body, you have lots and lots of different proteins. There's only one protein that your body would start breaking down more if you started to exercise, and you wanted to use that protein for as an energy source. The only protein who has enzymes that become more active and whose dam opens up are muscle proteins. So for us, we have all three of these molecules in here. And if we looked at catabolism first, so the breakdown, so say we're catabolizing them and we're going to use them for some process, like fuel energy for muscle movement for exercise. These molecules can be broken down into their component molecules. So what molecules are used to make them? Well, when they're broken down, they reform those molecules. These arrows that I just drew are pathways. So it wouldn't be just one chemical reaction. It could be multiple ones. These pathways, these rivers are always flowing. But again, there's dams on here that can open and close to increase or decrease these pathways. And what we'll find is very interesting. These pathways, these dams and how they're controlled, the chemicals that are around these enzymes influence, regulate these pathways, which is amazing. And even your thinking can speed up or slow down these pathways. Stress can influence these pathways. But that stress creates chemicals that influence how well the enzymes that are part of this pathway, how well they work. And it could be a good thing or a bad thing. It's amazing how your chemistry is controlled. But for right now, these rivers are always open, but the dams can open or close depending upon conditions around them. The component molecules of muscle protein, it's amino acids, and we'll learn more about those in the next chapter. The component molecules for triglycerides, which are fat and oil molecules, they're made from molecules that are called fatty acids and glycerol. And we'll see that chemistry a little bit later on. The component molecules of carbohydrates, well, glucose and fructose are monosaccharides, if you remember that. So they don't have component molecules. But glycogen, if you remember, is made from glucose. So here are component molecules: amino acids from muscle proteins, glucose from glycogen, and for triglycerides, fatty acids and glycerol. And we'll see all those structures a little bit later, but for right now, know that those are the component molecules. Those component molecules that are constantly being formed from these molecules, or maybe even from your diet, these component molecules can go this direction in the catabolic pathway. And if they do that, and this is, different pathways, these are different molecules, so that their pathways would be slightly different, but they can be broken down into waste molecules. And again, any fuel molecule that is completely catabolized, carbon dioxide, all those carbons in that fuel molecule or any molecule become carbon dioxide. There's one here shown as ammonia. We'll see that amino acids have a nitrogen functional group. They have an amine functional group within them. Those nitrogens that are in the amino acids become ammonia, and we need to go a step further. In you, they become urea. In fish, ammonia is the waste molecule. When these component molecules are catabolized, you harness chemical energy from them. And often there's molecules, chemical energy that the ones that take them that form, one of the molecules that can is called ATP. And we'll see that a little bit later. But there's other ones, they're taking chemical energy, they're taking electrons, so ATP. There's other ones that we'll see in this chapter like NADH. That's an acronym that we'll see a little bit later. But what's interesting, these molecules that took the chemical energy, they're products of this catabolic pathway, but they eventually become reactants in other pathways. As we'll see a little bit later, ATP, this molecule that took chemical energy from the fuel molecules, it's a reactant in the pathway for muscle movement. ATP was a product, it's a reactant to make your muscles move. So the chemical energy that was in one of these fuel molecules, that chemical energy can be used to move your muscles. Or these chemicals that have taken chemical energy from the component molecules can be used to build bigger molecules. So go the anabolic pathway. So if you wanted to build a protein, an anabolic pathway, product could be a protein. Or maybe a big molecule like your DNA, which is enormously big. Or maybe you were producing some fat molecules, but bigger molecules. The chemical energy that was in these component molecules is being used to make these bigger molecules. This is an overview of how energy goes from one molecule to another to build more molecules. If you didn't have an input or a storage of these molecules, you could never build these bigger molecules, or you could never make your body move. You always need an input of energy into a cell in order for it to survive. And that input of energy for you is coming from fuel molecules. And luckily for you, a multicellular organism, you have plenty of these fuel molecules stored in your body. You've heard that if you didn't eat but you're able to drink water, you could survive for more than a month. That's because you have quite a few calories worth of energy of these fuel molecules in your body. If you were a 75 kilogram person, I know it depends a little bit if you're male or female, but how many calories do you use per day? And let's just assume you were a student, so you're just studying all the time, you weren't exercising. And let's assume you're about 10% body fat. But let's say you're 75 kilogram, 10% body fat, which is pretty low, but not super low, but pretty low. You would use approximately 2,500 calories per day. Well, how much of each one of these fuel molecules do you have in your body? Well, as far as the carbohydrates, it's interesting at best, and it varies a little bit, you have about 1,600 calories of carbohydrates that could be used as a fuel molecule stored in your muscle and liver. Not a lot. If you're relying on this energy source and you didn't eat for a day, well, you would run out of energy and you wouldn't survive a day. It's not your stored carbohydrates that can allow you to not eat for 30 days or more. Okay, what about muscle protein? And again, it's only muscle protein that if you started to exercise, whose rate of usage would increase. Most of you have heard of insulin. It's a hormonal protein. You don't start using up more insulin for an energy source if you start exercising. It's only muscle protein that has their enzymes that become more active, that work better when you start to exercise. But how much muscle protein could you use and still be able to walk? I mean, you have a lot of muscle protein in your body, but there's only about 25,000 calories worth of muscle protein energy that you could use and you could still function. Which, you could last about 10 days if you're not eating off of just muscle protein. So, where's that enormously large supply that could last you more than 30 days? Well, it's your triglycerides. And triglycerides, again, are fat and oil molecules. And we'll be able to distinguish between a fat and oil molecule a little bit later. But triglycerides, you can find them in every cell, but most of them are stored in cells called the adipose cells. If you're this 75 kilogram person with 10% body fat, you have around 135,000 calories. Now, I know if you've taken nutrition, you may have slightly different numbers. You don't need to know it exactly, but know that it's well over 100,000. And it's this large supply, even if you're in really, really good shape with only 10% body fat, that's stored in your adipose cells that would allow you to live without food for quite a few weeks. And don't worry, I'm not recommending not eating, but this just shows you where your energy reserves are at and where the largest amount is at. Meaning you never run out of, if you're normal and healthy and you're eating, you never need to worry about running out of fuel energy. You have plenty in there. And these dams, again, they're always slightly open. It's kind of like the dams along the Spokane River. There's always some water that's flowing out, but they can be opened or closed. And when one type of fuel molecule starts to get a little bit lower, like your carbohydrates, these other dams would open up more. And we'll see how that happens, not on exam number 1, and kind of the basis on exam number 2. Exam number 3 will really see exactly, with specific examples and chemical reactions, how the rates of these chemical pathways are controlled. It's pretty amazing. Shown on this page is an overview of the chemistry, the pathways, and the cellular communication that we're going to learn for this quarter. This slide, there's nothing you need to know for exam number 1, or really exam number 2. You'll be applying it globally to the final exam. This diagram is also in your information packet on page 9. But it shows you the 3 main cells that we're going to be looking at when we look at metabolism. One of them is your liver cell, so that big circle, and don't make fun of my drawings, is your liver cell, the adipose cell, and your muscle cells. All these arrows in here are different pathways. There's also some key hormonal interactions, so these are chemical messengers that go between the cells. And we'll learn more about chemical messengers and hormones in some upcoming chapters. Also, some molecules that are produced in some cells and utilized in other cells, like lactic acid. This diagram shows you what cells, or what cell in particular it goes to, and that's the liver cell. Also, some chemicals that are insoluble in water, so they're insoluble in your blood. This diagram shows you how these molecules are transported from one cell to another. So fat, and most of us know that fat's pretty insoluble in water. If we're trying to clean our dishes at home, we know that. There's certain transport particles that are called lipoproteins, and there's acronyms for them that we'll learn about in exam number 4, like HDL, LDL, with something called VLDL, very low density lipoproteins, transport fat that's synthesized in the liver to your adipose cell. But again, this is more of an overview. It's in your information packet. I'm sure you'll glance at it a lot throughout the quarter as we present the information that's relevant to this diagram. A measure of the energy change that occurs in a chemical reaction, whether it's in a test tube or in your body, that measure is free energy. And hopefully you remember from general chemistry, free energy is given by a letter abbreviation G. And this is just energy. It's the chemical energy that a molecule has. Hopefully you also remember that this change of energy, this energy can be released to the surroundings, or the surroundings can provide energy for the chemicals to form products. If that change of energy, if energy is released to the surroundings, it's said to be an exergonic reaction. So remember endothermic, exothermic, that was for the enthalpy change only. Free energy change, it's exergonic or endergonic. If energy is flowing from the surroundings to the reactants to form products, it's endergonic. If an exergonic reaction happened in your cell, well, energy would flow to the surroundings. The surroundings would be your cellular fluids. The temperature of your cellular fluids would increase. If you remember, when you have a change, and you can have lots of changes in lots of different circumstances, but we designated a change in something with a triangle. So the change of energy, the change of free energy, was delta G. And in a chemical reaction, if free energy was lost to the surroundings, and this would be like you jumping from the third floor to the second floor, you would lose potential energy to the surroundings. That delta G, that change in free energy, is negative. These type of reactions are said to be thermodynamically favorable, and they're spontaneous. All of those mean the same thing. If you have a chemical reaction and delta G is a negative number, and there would be units, it's said to be thermodynamically favorable, spontaneous, and hexergonic. They all mean the same thing. If you have an endergonic reaction, and you have endergonic and exergonic reactions in your body, it takes energy from the surroundings. And taking energy could be taking kinetic energy from the surrounding molecules or maybe photons, but the products have more energy, more chemical energy than the reactants. This would be like if you're a reactant on the second floor and you go to the third floor, you have more potential energy than you did when you were a reactant on the second floor. The change in free energy, if it's an endergonic reaction, would be a positive number. And they're said to be non-spontaneous reactions and thermodynamically not. We forgot the not. Oh yeah, no, there's the not. Not thermodynamically favorable. So energy is energy, and you get the word free, and don't get hung up on the word free, but free energy, it's really the same thing as chemical energy. And the word free, you'd have to look at kind of the definition, but you have a chemical with a certain amount of energy. The free energy is the amount of energy that could be used during a chemical reaction to do work. So it's just called free energy. If you remember the plots of energy changes during a chemical reaction, we call these reaction diagrams. And you have a type for exergonic reactions and a type for endergonic reactions. So here are two plots. We haven't completed the reaction diagrams, but it kind of shows you the net change. In one type, the reactants that are a certain chemical energy, like a certain potential energy, and the products are at a lower free energy. So this would be like being on the third floor and going to the second floor of a building. From the starting point from the reactants down to the products, that's the change. And this is an exergonic type of reaction. This one, the delta G equals, and that's the slash means negative. This is a negative reaction diagram, the net change. The products lost energy relative to the reactants. Energy would be released to the surroundings. And the one over here, this is an endergonic reaction. The delta G equals a positive number. The reactants had to take in some energy to form products. This one is like going from the second floor to the third floor. And this just shows you the net chemical energy change for a reaction. If you remember, there's something called the activation energy. This is the energy required to break the bonds. Every chemical reaction has an activation energy. And sometimes it's, the bonds are stronger and sometimes they're weaker. The activation energy is for different types of reactants. Some can be greater, some can be smaller. This activation energy was abbreviated in general chemistry with an E. It was E sub A, and it equaled activation energy. And if you remember, when activation energy was part of this plot, the reactants had to gain some energy in order for bonds to be broken. And that energy, in your body, it always comes from collisional energy. But the reactants had to gain enough energy in order to break a bond. And we drew this kind of like a hill profile, an energy profile. And from the reactants, from there to the top of that peak, that E sub A, that's the activation energy. For reactants going to products in an endergonic reaction, you still get that hill profile, and you need energy to break the bonds. And from the reactants to that top of that hill, that's our activation energy. The reason why it goes back down, once you break the bonds, when bonds are formed to form products, energy is released. It always takes energy to break a bond. And that's because when you break a bond, you're pulling positive protons, atoms, away from their bonding electrons. There's an attraction between those two. And it takes energy to knock something that's attracted to something else. It's like pulling a north and a south magnets away from each other. That energy usually comes from a collision, and you may knock one atom away from this attraction to those bonding electrons. It always requires energy to do that. For this class and the type of questions you're going to have, what you need to be able to recognize is that this is an exergonic reaction for a reaction diagram, and this is an endergonic. Products are higher than reactants for endergonic. Products are lower in energy, chemical energy, than reactants for an exergonic reaction. Okay, so this is showing a reaction diagram for a single reaction. In your body, all the chemical reactions are linked together. So as soon as you form a product, it's going to become a reactant, and it has its own reaction diagram. And that's what's shown down here is a reaction diagram pathway for a chemical pathway in your body. And if we look at it, we're starting with A molecules and we're forming B molecules. So this would be one individual reaction diagram, but this B molecule can react and form something else. And this would be the first reaction, so we'll just say 1. Every time you see a hill, that's the activation top part of a reaction. The B molecules could form some C molecules, and this would be a second reaction. And then maybe those C molecules react to form some D molecules. So here's a third reaction. And maybe those D molecules form some E molecules. Here's a fourth reaction. So this diagram down here is a reaction diagram for a pathway in which there's 4 reactions. So maybe we have A forming B, B forming C, C forming D, and E is the last chemical that's part of this pathway. Some of the reactions in here are exergonic, some are endergonic. If we look at reaction 1, here's the reactants at a certain level. The products, the B molecules, are a higher free energy. That would be an endergonic reaction for reaction 1. Reaction 2, where B is here, and we go down to C, that's an exergonic reaction. Reaction 3, you should be able to tell, is endergonic, and D to E is exergonic. The net change of energy for this pathway, some of those reactions release energy to your cellular fluids, make sure cellular fluids get hotter. Some take energy, and the cellular fluids get colder. The net energy change for each reaction, so the delta G net, would be the sum of all the energy changes for each individual reaction. So it would be the delta G for reaction 1, plus the delta G for reaction 2, plus the delta G for reaction 3, plus the delta G for reaction 4. Add all those together and you would get some number, and you could give a delta G for the pathway. There's no numbers for these reactions, but you could look at where you start and where you finish, and where you finish is a little bit lower than where you start. Just by looking at this diagram, this delta G for this reaction is exergonic. Energy would be released to the surroundings. If those in molecule E, if they were maybe up here, higher than A, then that would be an endergonic reaction. It's kind of like going over to Seattle from Spokane. You know, when you go, you cross some mountain passes up and down, but when you're in Seattle, you're at a lower altitude. You know, you've lost some potential energy going from Spokane to Seattle. And that would be an exergonic trip. If you're going from Seattle to Spokane, you're going to end up at a higher altitude. Your net change would be an endergonic trip. If we go back to this slide where we showed glycolysis with the free energy change for each reaction, the ones that are negative, those are exergonic. Reaction 1 would make your cellular fluids, would make the temperature increase. Energy is being released to the surroundings, and the molecules would move faster. Some reactions, like reaction 8, that's a positive number, that's taking energy from the surroundings. It's taking kinetic energy from the surrounding cellular fluids. And if this one was happening, and if only that one was happening, the temperature of your cellular fluids would get colder and colder. But some are releasing energy, some are taking it in. To get the delta G net, all you need to do is add up these 10 numbers and get a numerical answer. Why don't you go ahead and pause the video and add all 10 of these numbers together and see what delta G net you get. Remember, some are positive, some are negative. Unpause the video when you're ready to check your answer. If you add all 10 of those numbers together, 3 are positive, 3 take energy from the surroundings. Most of them release energy to the surroundings. The delta G net equals negative 18.3 kilocalories per mole. This would be an exergonic pathway. In every organism, and you're an organism, every metabolic pathway is thermodynamically favored, which means the delta G for the pathway, that net delta G, is negative, which means energy is released to the surroundings. The cellular fluids get warmer when that pathway occurs. It doesn't matter if it's an anabolic pathway or a catabolic pathway, they're always exergonic. Shown down here are 2 different pathways, reaction diagram pathways. One is endergonic, one is exergonic. Why don't you go ahead and pause the video and state which one is exergonic, which one's endergonic, which one is a pathway that's spontaneous, which one would occur in your body. So go ahead and pause the video, unpause the video when you're ready to check your answer. So for the top one, look at the beginning and look at the end. The end products have a higher free energy. That delta G net is positive. That's an endergonic reaction. The bottom one, if you look at the initial reactants in the products, they're lower, they being the products, meaning the delta G net for that pathway is negative. This is an exergonic reaction. And like I said earlier, every pathway, anabolic or catabolic, releases energy to the surroundings. The net delta G is exergonic. And that would increase the temperature of your cellular fluids. If this pathway happened, and it was happening a lot, that would cool off your cellular fluids. And if you were doing a lot of those type of pathways, eventually your fluids would get so cold they would freeze. It wouldn't be a very good thing for your cellular fluids to freeze. Every pathway, even if you're building bigger molecules, is exergonic. So what if you're in a state of building proteins? You're growing a lot. It's exergonic, releases energy to the surroundings. But didn't I say earlier if we had an exergonic reaction, it takes energy? So remember back on this slide, we said amino acids, the proteins. In your body, well, anywhere in a test tube, it requires energy. Delta G is what's the net energy change to the surroundings. So let's discuss that just a little bit. Let's think about building proteins in a test tube versus your cells. You can take amino acids in a test tube, so amino acids, and you can make proteins. If you just let this reaction happen, it requires some heat. This is an endergonic reaction, and the energy could come from the kinetic energy of the surrounding molecules. But that's not what your body does. What your body does, instead of using heat energy, so the surrounding kinetic energies of the molecule, what your body does is it uses molecules like ATP and amino acids, and it's a little more complicated than this, to make proteins. It uses chemical energy. And some of that chemical energy in ATP goes directly to the proteins. Some of it is lost as heat. And that heat loss on the product side makes it exergonic. So in both cases, to make a protein from amino acids, you need energy. You could get energy from the kinetic energy, which your body doesn't. This one, the surrounding kinetic energy is used to make it in the temperature we get colder. Using chemical energy, like what's done in your body, it's not going to decrease the temperature of your cellular fluids. And it makes it so that pathway is exergonic. Every pathway, whether it's anabolic like this or catabolic, releases energy to the surroundings. The delta G isn't the energy input, it's the net change of energy. The net change of energy to the surroundings or into the reactant and product molecules. which means that delta G tells you the heat flow from your chemical reaction to your cellular fluids. So as you go along, just remember, as I present all of these topics, and you're wondering what type of questions I could ask, use your study guide with all those wonderful practice problems, and use the keys that are also posted to guide you on the type of questions that are going to be asked. They're extremely beneficial. Which leads us to another topic called coupled reactions. And new word, and it may seem complicated at first, but again, look at the practice problems. You'll get it, I promise you. Coupled reactions is a concept presented in biochemistry. The concept is that you have two reactions that could happen with or without each other in a test tube. When you're an organism, these two reactions are coupled together through the act of an enzyme. And you make some sort of reaction that's very unfavorable. It's highly endergonic when it acts alone, but coupled with another reaction, it becomes exergonic. It's basically you have two reactions and you're figuring out a net reaction and a net delta G. So let's go over this concept and make it more clear than what I just said. Okay, the reaction that I'm going to look at first is glucose reacting with a phosphate. If you remember from the glycolysis set of pathways, the very first thing that happens to glucose is that a phosphate is attached. This happens in any organism. Glucose in the blood is glucose. The first thing that happens to it chemically in your cell is that a phosphate gets attached. And this can happen with an enzyme or without. This could happen in a test tube. You can have glucose that reacts with a solvated phosphate, so glucose plus phosphate, and you can form glucose 6-phosphate. And this formula, that represents phosphate. This can happen in your cell. You have lots of solvated phosphate in your cell. This could happen in a test tube. In this reaction, you also produce water, so plus water. The thing about this reaction, though, it's highly endergonic. The delta G is positive. This is the first reaction that happens to glucose in any cell in your body, any cell in any organism. It's kind of a bad thing if this reaction is unfavored. It makes it harder to react. The transfer, the attachment of phosphate to carbon-based molecules, it occurs throughout your pathways. It's a primary reaction that happens in anything that we consider alive. And depending upon phosphate getting attached to a carbon-based molecule from solvated phosphate, and the fact that it's endergonic, isn't very favored. If there's some other way to get a phosphate attached to a carbon-based molecule, that could be very beneficial. Attaching phosphate makes the bonds incredibly weak. It makes carbon-based molecules more reactive. And we'll discuss that in just a little bit. But it's definitely a primary reaction, attaching a phosphate. So, what could an organism do? Is there any other way to get a phosphate? And there's molecules that contain phosphate, and molecules, acronyms you've probably heard of, like ATP. We'll see ATP in the next few slides, and we'll see its structure and understand it a little bit better. But for right now, we'll just know that ATP exists. It stands for adenosine triphosphate. It has a adenosine molecule with 3 phosphates attached, and its acronym is ATP. ATP can, if you put it into water, can react with water and lose one of its phosphates and form, a solvated phosphate ion. What's left over on the adenosine molecule is just a diphosphate, so ADP plus a hydrogen cation. Okay, we'll see this reaction in just a little bit, but for right now, you'll just have to trust me that it's a highly exergonic reaction. So somehow if we could bring these two reactions together, and that's the way the word coupled comes, could glucose get its phosphate from ATP instead of from a solvated one? That would be nice. A high energy molecule like ATP, you're easily losing a phosphate to glucose. That would be a lot easier. A coupled reaction is really the net reaction of two reactions. So we're eventually going to get the net reaction from these two, and it's called the coupled reaction, the net reaction. So if we call this reaction 1 and this reaction 2, and if we figured out the net reaction of those two, That's the coupled reaction. And in biochemistry, whenever you have a coupled reaction, again, one of them is endergonic and one of them is exergonic. What brings those reactions together is an enzyme, and we'll see how enzymes work a little bit later. But to figure out a coupled reaction, all you have to do is figure out the net reaction of two reactions. So let's think about it. We sort of did it halfway or partway for 10 reactions in glycolysis. How do we get the net reaction for these two? So remember what we said, if something's produced then used up, you don't see it in the net reaction. If something's a product and it's a reactant somewhere else, it gets canceled out. So what do we see in these two reactions that would cancel each other out? Water's produced in reaction number 1, but it's used up in reaction number 2. So even though it's part of it, it's not going to be seen in the net reaction. Phosphate's used up in reaction 1, but it's produced in reaction 2. So in the net process, it's not going to be seen in the net reaction. Okay, that's it for things being canceled out. As far as writing the net reaction, so it's still a chemical reaction, so there's an arrow. Anything that's a reactant in both reactions would be a reactant in the net reaction. So glucose and ATP, those are the two reactants. So on the reactant side of the net reaction, it doesn't matter which one you draw first, but glucose plus ATP. On the product side, we see glucose 6-phosphate, ADP, and a hydrogen cation. So there's 3 products. So glucose 6-phosphate plus ADP plus the hydrogen cation. So this is our net reaction. It's also our coupled reaction. A couple things for a note. When we have phosphate, and here's the formula for phosphate. There's abbreviations in biochemistry that I'll use and are used in your book. Phosphate can be abbreviated as a PI, so that equals phosphate. And just note, if you were curious about its structure, you should kind of remember it from 122. There's 2 negatives, so the 2 negatives on the oxygens and the OH. And also remember, not super important, but remember the hydrogen cation. That's an abbreviation for the hydronium ion. Sometimes I'll write the hydronium ion, sometimes I'll write the hydrogen cation. In reality, there's no hydrogen cations in an aqueous solution, but it's written that way just as an abbreviation. And remember, acidity is related to the hydronium ion concentration. We'll be talking about that in upcoming chapters. When ATP is produced in your body, it's produced in every cell. When it's produced in a chemical reaction, it's taken chemical energy from some other molecule. ATP is then used to deliver chemical energy to another molecule. That's the main purpose of forming and using up ATP, is to take chemical energy and to deliver chemical energy. When ATP is synthesized, it's made from adenosine diphosphate, so ADP. What's true about ATP or ADP or any or most chemicals that are in your cell, your body tries to maintain a constant concentration. So let's draw a lake of ATP. It's at a certain chemical energy, certain potential energy. It's made from ADP. I'm going to make its lake level a little bit lower, so ADP. But it requires chemical energy to make ATP, so we'll just say energy, E for energy. When ATP is made, it's used up and it reproduces ADP. That's the most of the time. Sometimes it makes other chemicals too, like AMP, but we'll see those on the next page. But that's the sole purpose. ATP takes chemical energy when it's being made, it gives it away, so we'll kind of, the energy, it goes and does something. It could be used for muscle movement or maybe to build bigger molecules like proteins. During the day, you use about 50 kilograms of ATP per day. So 50 kilograms of ATP is used. If the strategy of the body is to keep that concentration constant, that means about 50 kilograms of ATP is produced per day. So if you started exercising and started using a lot more ATP maybe per hour, the dams would open up and you'd start producing a lot more per hour. We'll see that chemistry a little bit later, starting with exam number 3. For right now, let's look a little closer at the structure of ATP. Its structure is very similar to other bigger molecules in your body. ATP is something that's called a ribonucleotide. The molecules that are used to make ATP are also used to make your DNA and your RNA. This portion right here, the whole molecule is ATP, but this portion that I just circled, it's a molecule that's called adenine. Adenine is used to make ATP. Down here is a sugar that you should recognize from Chemistry 122. That's ribose. These two molecules together make something called adenosine. And here we have 3 phosphates connected together. That's triphosphate. So we have adenosine triphosphate. We'll see the structure of adenosine diphosphate and adenosine monophosphate on the next page. But if it was adenosine diphosphate, then one of these phosphates would be knocked off. Monophosphate would just have one phosphate attached to the adenosine. One of the primary chemical functions of ATP is to transfer attach a phosphate to another carbon-based molecule. It's the one that we'll see react the most when we look at our chemical reactions a little bit later. However, there's other ribonucleotides that do exactly the same thing. These other ribonucleotides, guanosine triphosphate, GTP, uridine triphosphate, UTP, cytidine triphosphate, CTP. We'll see GTP in our chemical pathways a little bit later, and UTP. We won't see them nearly as much as ATP, and they don't get the love like ATP does in your biology books, but they're incredibly important. They're also made from molecules that are used to make other bigger molecules like your DNA and RNA. We'll see that kind of as a common theme. There's a group of molecules that are used a lot to build other bigger molecules. Shown on this page are 4 different adenosine phosphate type of structures, and I'm pretty sure you could label them with the acronyms. One of them is adenosine triphosphate, adenosine diphosphate, and adenosine monophosphate. The 4th one is called cyclic AMP, or cyclic adenosine monophosphate. We'll see all 4 structures in our chemical pathways as we go through this course. What I expect you to be able to do on exam number 1, if I gave you these structures, you should be able to label them with the correct acronyms. I don't expect you to spell out ATP, but I do expect you to be able to recognize this top structure on the left as ATP. There's 3 phosphates, 3 Ps. Over here is adenosine diphosphate, 2 phosphates connected together. And here's adenosine monophosphate. Here is a structure, you see that cyclic structure. This is cyclic AMP. And sometimes it's abbreviated as CAMP. When we get into the chapter that discusses hormones, we'll see that this cyclic AMP is a molecule that's produced due to a hormonal response. But we'll save that fun for a little bit later. For this chapter, for exam number 1, I expect you to be able to recognize these. There's 3 other structures we'll present before the end of this chapter that I expect you to be able to label. Not draw, but be able to label. One last note for these chemicals. So you have all of these chemicals in every cell. If you summed up all 4 of their concentrations in your cell, it's relatively constant. That's because, if ATP reacts, it either forms ADP or AMP. If one concentration, if its concentration goes up, it means the other ones are going down. If ADP concentration is going up, it must mean ATP's concentration is going down. Okay, I just wrote really quickly. Let me read what I wrote. So total concentrations of ATP, ADP, AMP, and cyclic AMP is constant in a cell. If for some reason ATP concentration started to decrease, like you started to exercise and it started being used up a lot to make your muscles move, then the concentrations of these other adenosine molecules would increase. If one goes up, the others, well, ATP is the one that is by itself, but if ATP's concentration increases or decreases, those other three would have the opposite increase or decrease. On this page, we're going to discuss the energy that is released, the chemical energy that's released when ATP reacts and a phosphate is cleaved. So here's the structure of ATP, except I'm not showing the adenosine portion, just the triphosphate portion. So I'll just write ATP here. If this bond is cleaved and you form a phosphate, and this could happen from a reaction where water collides and breaks that bond, what's formed is adenosine diphosphate, ADP, plus a free phosphate, and I'll just write PI, remember that can stand for phosphate, plus the hydrogen ion. The free energy change for this process is negative. And don't worry if we don't get the units kilocalories per mole, but it's negative. Energy is being released to the surroundings. There's stored free energy in adenosine triphosphate. So remember kind of our map if we went back to the correct one. Here's adenosine diphosphate. It takes chemical energy, and we're storing chemical energy in the form of adenosine triphosphate. When that reacts, no matter what the reaction is, it gives off energy when that phosphate is cleaved. And that's why we see a negative delta G. Energy is being released to the surroundings. If this reaction happens in water, the energy released just goes into the water and heat's produced, and the water's temperature would increase. If this phosphate, instead of just becoming a solvated ion like it is in this chemical reaction, if this phosphate is transferred to a carbon-based molecule like the glucose, then the chemical energy in ATP attaching that phosphate to a carbon-based molecule is transferring some of this chemical energy to the other molecule. So for this reaction in water, this delta G, that's telling us how much energy is going directly into water. It would be nice if some of this energy could be transferred to another molecule. And that's what ATP does when it transfers one of its phosphates to another molecule. And that's basically what this statement is saying. Said, I use a bigger word like liberated. This reaction that I'm showing here is also in your information packet, and the delta G for this reaction is also given. On page 3 of the information packet, ATP plus water forms ADP plus phosphate plus a hydrogen ion, and the delta G a negative 7.3 kilocalories per mole. If you remember some of your principles in chemistry, general chemistry, if you reverse a reaction, the energy, the delta G, it changes sign. So in this first reaction, we have ADP plus phosphate plus the hydrogen ion being formed. If you reverse the reaction, ADP plus phosphate plus a hydrogen ion, to form ATP plus water, the delta G, it's the same number, but it's positive. One reaction releases energy, the reverse reaction takes in energy. One of them is exergonic, one of them is endergonic. The top one, this is an exergonic reaction. The bottom one is our endergonic reaction. So here are the two reactions. I'm just rewriting them from the information packet. One's exergonic, one's endergonic. When we start looking at coupled reactions in more detail, and we're going to start with more detail on the next page, we're not going to look at every possible type of coupled reaction. The ones we are going to look at, one of these two reactions will be part of it. So remember, a coupled reaction, there's an endergonic reaction and an exergonic reaction. You always have both of those, one of each, in a coupled reaction. And again, for everyone we're looking at, one of these two reactions will be part of it. So I highlighted it down here. For all the coupled reactions that we will examine, one of these two reactions and one of these two, that one or that one, not both, only one of them, one of those two reactions will be part of it. The two different types of coupled reactions we'll look at both involved ATP. In one type, ATP is being used up. It's going to be a reactant. In another type of coupled reaction that we're going to examine, ATP is being produced, and it'll be a product. Okay, let's go examine coupled reactions a little more thoroughly. On this page, we're examining a reaction that involves fructose. And if you were able to recognize this structure as being fructose, you should pat yourself on the back and go tell your organic chemistry instructor. In this reaction of fructose, the very first thing that happens to fructose when it enters a cell is that a phosphate is attached. And this is just like any other carbohydrate, and like glucose, a phosphate is attached when it enters the cell. It's fructose in the blood, it's glucose in the blood. When it enters the cell, there's enzymes in there that catalyze the reaction to attach a phosphate. When fructose reacts inside your cell, it forms fructose 1-phosphate. And if you could recognize that that's the 1 carbon, pat yourself on the back another time. Again, this reaction, it's highly endergonic, requires a lot of energy, but it's needed. Attaching a phosphate, it does more than just provide chemical energy. You're attaching 4 oxygens. And if you remember your general chemistry and a little bit about oxygens and electronegativity, you're attaching 4 atoms that are the second highest electronegativity atoms. And remember what electronegativity means. It means those atoms have a strong pull for electrons. And it's not just the electrons that are bonding to them, they have effect on neighboring electrons. These 4 super suns that are here weaken the bonds in the other parts of the molecule. So attaching the phosphate makes the bonds weaker in the carbon-based molecule. And you need these bonds weaker so that the chemistry that happens downstream from this point, it's easier to happen with the bonds weaker. So somehow, if we could get a phosphate attached to fructose without worrying about phosphate that's just solvated in your cellular fluids, how could we do it? Oh, maybe that molecule. On the next page, we're going to see the coupling process in which ATP is used to attach the phosphate, just like we did for glucose. On this page, we're showing you the coupling process for that fructose reaction. So here's the reaction we had on the previous page. We'll just call that reaction number 1. It's the reaction that's endergonic. And again, when we have coupled reaction, one is exergonic, one is endergonic. The exergonic reaction that we have, and this is reaction number 2, is the ATP reaction in which ATP forms ADP. That's the exergonic reaction. Remember, all the coupled reactions we're looking at, either ATP is going to be a reactant or it's going to be a product. One of those 2 reactions. The coupled reaction is the net reaction of both of those. And remember, the delta G net would be the sum of the 2 individual delta G's for both reactions. Why don't you go ahead and pause the video and write the net reaction and get the net delta G for the process. Unpause the video when you're ready to check your answer. So remember, for the net reaction, if something's produced and then used up, it's not seen in the net reaction. So water's produced, but then it's used up, so we don't see it in the net reaction. Also, phosphate, it's on both sides of the equation. It's a reactant and a product, so we don't see it in the net reaction. To get the net reaction, this is a reactant, that's a reactant. They're both the reactants in each reaction, so those are the reactants in the net reaction. And there's 1, 2, 3 products, and we see 1, 2, 3 products. The delta G net, just add the 2 delta G's from each reaction. Remember, one's negative. The delta G net for this coupled process is negative 4.0 kilocalories per mole. So what does this mean exactly? If ATP lost a phosphate, it releases negative 7.3, that much energy to the surrounding as heat. When it's coupled to this process, if that phosphate is directly transferred to fructose, it still loses some energy. That delta G net being negative 4 means that's energy going to the surroundings. But some of that 7.3 went into fructose 1-phosphate as chemical energy. Okay, one last note about coupled reactions. I know we have them separate like this, but what exactly is happening in your body? Well, what's happening in your body when fructose enters the cell, it reacts directly with ATP and it gets a phosphate attached. And there would be some sort of molecule, some enzyme, some big protein that brings those two molecules together. And we'll have to learn about enzymes in a couple chapters, but somehow that phosphate gets connected and makes a phosphate ester to carbon number 1, not the other carbons. On the next page, we're going to look at a coupled reaction in which ATP is being produced. There's a variety of reactions in your body where ATP is produced, and we're going to see that coupled reaction, that net reaction on this page. So here's one reaction, here's another one. One of them is exergonic, one of them is endergonic. This is a reaction that happens in a pathway that we gave an example as earlier, and it's called glycolysis. And this is the number 10 in that pathway, and it's the one in which pyruvate is being produced. If you add these two reactions together and get the net reaction, then that's the coupled reaction, and hopefully it agrees with our glycolysis reaction that we saw back on slide number 3. It should be just like reaction number 10. So what I want you to do is pause the video and get the net reaction for this and get the net delta G. When you do write it out, you don't have to draw the structures even though I showed it. You can just write the names of the molecules. So you don't have to draw the structure of pyruvate. You can just write the name of pyruvate down here. Unpause the video when you're ready to check your answer. Okay, let's go ahead and go over our answer. So as far as what gets canceled out, water is used up in reaction number 1, but it's produced in reaction number 2, so we don't see it in the net reaction. Same thing for phosphate. And that's it for canceling. So phosphophenol pyruvate plus ADP plus a hydrogen ion forms pyruvate plus ATP. The net delta G for this process is negative, so it's the net reaction is exergonic. And again, one reaction is exergonic, one is endergonic. So when phosphophenol pyruvate reacts, and if this just happened in water, it releases that much energy as heat to the water. When it's part of a net reaction and it transfers a phosphate to ADP, some of that chemical energy that's in phosphophenol pyruvate becomes part of ATP. The negative 7.5 that's remaining, that's what goes off as heat. Whenever you see a delta G and it's negative or positive, that's the heat flow to the surroundings, either to the surroundings or from the surroundings. Okay, one more thing to reiterate. As I mentioned earlier, when we have coupled reactions, one of the reactions will either be ATP as a reactant or ADP as a reactant. This reaction here, if we go to our information packet, that's this reaction. When ATP is being produced, this is the reaction that's part of the coupled reaction. If ATP is being used up, then it's the top one. And lastly, another page about coupled reactions, we're going to make your mind think a little bit. On the previous examples, we gave you the two reactions and you had to come up with a net reaction. This time, we're doing it a little bit differently. You're given the net reaction, and somehow you have to figure out what are the two reactions that when they're added together give you this net reaction. So you're given the answer and you have to work backwards. It's very analytical, just kind of like a math class. Shown here are steps to follow that'll allow you to get these two reactions. Okay, let's practice. So we have this reaction. Step 1, we need to determine which ATP reaction we're going to use. We're going to use one and only one. And what you should do is when you look at the net reaction, remember it's the sum of these two, what side is ATP on? If it's a reactant, if it's on the reactant side, then you should use the reaction in which ATP is a reactant, which would be this one. If ATP was a product in the net reaction, then you should use this bottom one. In our example, it's on the reactant side. So this is the reaction that we should use. This reaction, it's negative, so it's the exergonic one. And again, remember, one of these two reactions, and only one, is going to be part of our coupled process. So if we go back to our lecture notes, reaction 1, and it doesn't, really doesn't matter what you call 1 or 2, but one of the reactions is this ATP reaction. The next part is to try and figure out what is this second reaction. That's steps 2 and steps 3. This reaction that we're creating, it's a chemical reaction, so there's an arrow, but when these two are added together, it needs to equal this. We have the first reaction, we need to figure out the second one. Okay, as we look at step 2, basically what it says, any chemical that's in the net reaction, so any chemical down here that's not up here, we need to write down here. Because remember, when these two are added together, it needs to equal this. So what's in the net reaction that's not in this ATP reaction? If we look on the reactant side, this 3-phosphoglycerate is in the net reaction. It needs to go in reaction number 2. ATP, well, it's up here, so we don't need to add it. On the product side, what's on the coupled reaction that's not in the ATP reaction, this 1,3-bisphosphoglycerate, it's in the coupled reaction. It needs to go in reaction number 2. ADP, well, it's already here, so we don't need to add it. And the last step, anything that's in the ATP reaction, so anything up here that's not in the net reaction, we need to cancel out using reaction number 2. So cancel out. So let's first decide what's in reaction 1 in the ATP reaction that's not down here in the net reaction. So if we look up here, water's not in the net reaction, so it needs to be canceled out. What else do we have? Well, phosphate. Phosphate's in reaction 1. Somehow we need to put it in reaction 2, so when reaction 2 is added, they cancel each other out. Also, we don't see a hydrogen ion down here, so it needs to be canceled out. So canceling out, what does that mean? Well, water is a reactant in reaction number 1. To get it canceled out, it needs to be a product in reaction number 2. So plus H2O. On reaction number 2, we also need to cancel out phosphate in the hydrogen ion. Phosphate's a product in reaction number 1. To get it to cancel out, it's a reactant in reaction number 2. Same thing for the hydrogen cation. To cancel it out, it needs to be a reactant in reaction number 2 since it's a product in reaction number 1. Okay, just to make sure we're doing things correctly, it's always good to check your workout. So here's our 2 reactions, 1 and 2. You just make sure things cancel out and you come up with this reaction. So hydrogen ion, reactant, and product cancels out. Phosphate and phosphate, they cancel out. H2O and H2O cancels out. If we added these together, we have ATP and the 3-phosphoglycerate as a reactant. That's what we have there. On the product side, we have ADP and that 1,3-bisphosphoglycerate as a product. 2 reactants, 2 products. It worked out. Okay, it sounds like, to me, with all this information, it's time to practice, so we get it down better. If we go to our study guide and look at all the practice problems, and I'm just going to show you this so you can see how valuable it is. But within the study guide, here are practice problems. That kind of stuff looks familiar. Oh, these things kind of look familiar. And here's where we start seeing coupled reactions. Those look pretty easy. Just add those together. 7, 8, if you look at those, you're given the net reaction and you're trying to come up with the 2 reactions that added together give you that net reaction. So it's good to practice to reinforce what you're learning. And remember, the keys are posted on Canvas. It's good to know if you're doing your problem right too. And remember, these problems are just for practice, But those exam questions are going to look very, very similar to these practice problems. In this last part of the chapter, we're going to look at some of the catabolic pathways of your fuel molecules, and we're also going to look at some biological molecules, some coenzymes that are found throughout your metabolic pathways. On this slide, we kind of see a general overview for the pathways that are used to take the chemical energy from your fuel molecules. So we kind of showed this earlier, but without these big arrows. But you can get fuel molecules from your diet, and when you look at lipids, this is the same thing as something like a fat, and they're also called triglycerides. You can also get carbohydrates and get energy source like glucose, sugar, starch, and proteins. What's interesting, when these fuel molecules are catabolized, they're different molecules to begin with, but when they're broken down into smaller molecules and chemical energy is taken from them, you see how all these arrows kind of end up in the same general flow of pathways? The molecules, even though they start out different, they all become some very, very similar molecules. And when that happens, the enzymes that are used downstream, down here, you can use the same enzymes eventually to finish catabolizing fuel molecules. They start out differently, but they all flow into the one big river. Think of these as, that's a river, that's another river. A molecule that can be produced from the breakdown of any fuel molecule, it's found at the heart of these pathways, is called acetyl coenzyme A. And we'll see it and look at it in more detail on the next page. But when I say it's produced, what does that mean exactly? Well, the carbon atoms in this acetyl coenzyme A, they're coming from the carbon atoms from these fuel molecules, from proteins, carbohydrates, or fat molecules. A couple other molecules that we're going to talk about in the last few pages of this chapter, the chemical energy in these fuel molecules, and we talked about forming ATP, there's also other molecules that can take that chemical energy. The acronyms for these molecules that are formed, NADH, FADH2. We'll see those on the last few slides of this chapter. The last part of this chapter, we're going to look at molecules called coenzymes. These are molecules used by enzymes, and without these molecules, the enzymes don't work. Coenzyme A is the first molecule we're going to look at, and it's this coenzyme that is used to form acetyl coenzyme A. Shown down here is the structure of coenzyme A, and I don't expect you to be able to draw this structure, but this is one of the 7 molecules that you need to be able to recognize and be able to label. So remember, it's those ATP ones that you have to be able to recognize and label. This is another one, and there's 2 more beyond this. If you look at coenzyme A, it actually looks kind of similar to ATP, or maybe ADP. Here's the adenine molecule that's being used. There's the ribose. We have adenosine. And then there's a diphosphate. It's pretty interesting. Similar structure, similar molecules to ATP and ADP and AMP. Okay, but it's different. What makes it a little bit different are 2 other molecules that are also part of the structure. One of them is a vitamin. And I don't expect you to know the vitamins or what B vitamin is, but it's B5. If you've taken a nutrition class lately, and I know a number of you have to take nutrition, often they'll tell you where the vitamins, what pathways are they used in. And anywhere coenzyme A is used, if you want coenzyme A, if you want to be able to have it, you need to have vitamin B5 in order to synthesize it. Okay, the reactive site, the place where this coenzyme reacts, it's a thiol functional group. This molecule that has this thiol group is made from an amino acid, cysteine. We'll talk about amino acids in an upcoming chapter. Well, in the upcoming chapter. But this thiol group, it forms a thiol ester, and often it connects to a 2-carbon molecule called an acetyl group. So 2 carbons are actually cleaved from some carbon-based molecule, and this acetyl group would attach to that sulfur and make a thiol ester. Sometimes there's more than 2 carbons. It could be an R and carbonyl group, and you still make a thiol ester with the coenzyme A. When there's multiple carbons there, it's called an acyl group. We'll see acetyl coenzyme A throughout the metabolic pathways we look at, and we'll also see acyl coenzyme A. If there's an acetyl group attached to it, and it being the coenzyme A, it's called acetyl coenzyme A. Okay, when you label coenzyme A, you can write it just CoA, and that's the way I'll write it most of the time. Sometimes it's written this way, HS coenzyme A. And this is just showing that's where it's reacting at. If there's an acetyl group attached, usually I'll just write acetyl with an L, coenzyme A. Here in this example, they've also shown, well, I've also shown the acetyl group attached to the sulfur. Whatever makes sense to you, whichever is the easiest, I'll do that one in that form mostly. As far as the purpose and the function of coenzyme A, its main purpose is to take 2 carbons from some other molecule. And if we want to draw lakes and stuff like that, there's a concentration of coenzyme A in your cellular fluids. When it reacts, it forms acetyl coenzyme A. Once it forms acetyl coenzyme A, its sole purpose is to give that acetyl group to some other molecule and reform coenzyme A. So if coenzyme A's concentration decreases, it means acetyl coenzyme A's concentration is increasing. This is its main purpose. When it reacts as a coenzyme A, it reacts with some enzyme, and it would get a 2 carbon attached, and then it would diffuse away and go to another enzyme, so a different enzyme. The acetyl coenzyme A would go to a different enzyme. And note, the coenzyme A, there isn't just one enzyme it reacts with. There's a variety of coenzymes. There's a variety of enzymes that coenzyme could react with. And we'll see those reactions throughout as soon as we start getting into chapter 3. Okay, now we have 2 more coenzymes to talk about. The next coenzyme we're going to talk about, it has an acronym NAD and a plus. The plus is because it's a cation. It's called nicotinamide adenine dinucleotide. It looks very similar to coenzyme A, ATP. It has that adenosine backbone, just like those, another diphosphate. What makes it different is the vitamin that's being attached. And this is vitamin B3. This particular coenzyme, its chemical properties, they're different than coenzyme A. This particular coenzyme, it has a chemical property, it's a good oxidizing agent. It takes electrons from carbon-based molecules. Remember, if it takes electrons, it's taking chemical energy from a carbon-based molecule. Okay, let me read what I just wrote here. NAD+, it's an oxidizing molecule, and things that take electrons are called oxidizing agents. It takes electrons when it chemically reacts and hydrogens, and I know you can't read that part, from carbon-based molecules. If it takes electrons, it means it's taking chemical energy from the molecule it oxidizes. And we'll see that reaction on the following page. But one thing to note, this structure that's shown here, this is the 6 of 7 structures that you need to be able to recognize. So this structure, just like this, may be on the exam, and you may have to label it with its acronym. You don't have to remember, and I'll mostly just say the acronym. This is the last time I'll say nicotinamide adenine dinucleotide. That's a lot to say. That's why I use the acronym, and you can too. But again, it's interesting. This coenzyme, just like acetyl coenzyme A, is formed from ADP. There's a lot of similar molecules and similar structures that are used throughout your metabolic pathways, and this is another one. And probably the last coenzyme we talk about will be pretty similar too, but we'll have to wait and see. So here's a chemical reaction with NAD+, and most of the time I'll just say NAD, but it's only showing the vitamin portion, so here's an R there, where the reactive site is at. If we looked at our diagram here, we're just showing that part. Most of the molecule equals that R. When NAD, or NAD+, whatever you want to say, when it reacts with a carbon-based molecule, it always takes 2 electrons. And it takes 1 hydrogen away from the carbon-based molecule. That carbon-based molecule may lose more than 1 hydrogen atom when bonds are broken, but when NAD reacts, 1 hydrogen bonds to that molecule and you form NADH. NAD+, this is a coenzyme. It's an oxidizing agent. What forms NADH is also a coenzyme. NAD will react with some enzyme, take electrons, diffuse away. NADH that forms, it will diffuse and go into another enzyme, and it's going to give those electrons, it's going to give that chemical energy to another molecule. So let me go over what I just wrote. So NAD+, it's a coenzyme. It's something that oxidizes. It's an oxidizing molecule. NADH is also a coenzyme. Sometimes it's called a reduced coenzyme. It's carrying those electrons and going to give it to some other molecule, so it's said to be a carrier of energy. In fact, it has a lot more energy to give than one ATP molecule. It's also called a reduced coenzyme. And reduced, I know that's a general chemistry term, but it gives electrons to some other molecule. And when this reaction happens, it's kind of like coenzyme A to acetylcoenzyme A. You have a set amount of NAD and NADH molecules in your cellular fluids. The whole function of these 2 molecules, NAD will react, and we'll put a plus there, and form NADH. It does this by taking 2 electrons and a hydrogen cation. So we'll kind of draw an arrow. NADH will react and give its electrons and that hydrogen cation to somewhere else. So 2 electrons plus a hydrogen cation. Somewhere else could be another molecule. But NAD reacts to form NADH. NADH reacts to reform NAD. And you have that circle. It's kind of like ATP, ADP, ADP, ATP. A very common reaction for NAD with your biological molecules is to oxidize alcohol functional groups in your carbon-based molecules. I'm going to draw some sort of generic alcohol group, and NAD will be a reactant. I want you to go ahead and complete this reaction. So here's our alcohol. There's NAD. There's our arrow for our chemical reaction. Go ahead and pause the video and write the structures of the products. So remember, that's just our generic R group. Unpause the video when you're ready to check your answer. So we have a primary alcohol and an oxidizing agent. And I know you did this reaction in your organic chemistry class, and your teacher would be so proud of you if you're able to complete it. But when this reaction happens, you form an aldehyde. One of the hydrogens that were lost from that alcohol, one of them bonds to NAD to form NADH, and the second one forms the hydrogen cation. 2 electrons from this alcohol are part of that reduced coenzyme. And those 2 electrons would eventually react somewhere else. Most chemicals that are products become reactants somewhere else. And that's true with these coenzymes and reduced coenzymes. On this page is another coenzyme. This coenzyme is also an oxidizing agent, just like NAD. It takes electrons from carbon-based molecules. It's another carrier of free energy. It's called flavin, made from riboflavin, adenine dinucleotide. Its acronym is FAD, and again, just like NAD, that's all you need to say for this class. But if we look at it, there's the adenosine portion, there's the diphosphate, so it's made from ADP, and what makes it FAD is the vitamin that's used, so B2. So if you knew what pathways FAD was used in, which you will later, you would know that B2 is a pretty important vitamin for that metabolism. And again, remember, we're talking about fuel metabolism for this quarter. There's some B vitamins that are pretty important because they're used to make molecules that are part of those pathways. The part where electrons are taken is this upper 3-ring structure, and on the next page where we show the chemical reactions, this portion down here will be our R group. So here's FAD, there's the R group. When it reacts, it also takes 2 electrons, just like NAD. But this time, instead of just bonding 1 hydrogen atom to the coenzyme, 2 are bonded. And that's why it becomes FADH2. So FAD is a coenzyme, FADH2 is a coenzyme, but FADH2 is called a reduced coenzyme. And that FADH2 that's formed, it's going to give its electrons that it took from some carbon-based molecule. It's going to take that chemical energy that it took, that it acquired, to some other carbon-based molecule. FADH2 is also a carrier of free energy. And just like NADH, it has a lot more energy than ATP. And just like NAD, the sole purpose of FAD is to take electrons to form FADH2. So we can look at our little flow. The FADH2 that forms, its sole purpose is to give electrons and reform FAD. So FAD plus 2 hydrogens plus 2 electrons forms FADH2. When FADH2 reacts, it reforms FAD, and it gives away 2 hydrogens and 2 electrons. Okay, let's look at a reaction that involves FAD. It's an oxidizing agent. It's taking electrons. A chemical reaction that FAD, a reaction that it does, it converts a carbon-carbon single bond into an alkene functional group. And I know if you used the organic chemistry book that's used at our school, you didn't look at this chemistry. You looked at the opposite chemistry. You went from an alkene to a carbon-carbon single bond, but I'm pretty sure you didn't do the opposite. But let me just give you some sort of generic structure. So we have 2 R's, and those R's could be any sort of structure. And it's this single bond right here that's going to become an alkene functional group. But 2 hydrogens, 2 of those carbon-hydrogen bonds are broken, and we form FADH2, and 2 electrons came along with those 2 hydrogens. Instead of having a hydrogen cation, they both went with the coenzyme. And what's left over is a carbon-based molecule with a double bond between those carbon atoms, and there's still a carbon-hydrogen bond on both of those carbon atoms. So there we go. We have our alkene structure. So again, just for flashcards for you, remember there's 7 carbon-based structures that you need to be able to recognize on the exam. Those 4 adenosine phosphate molecules and these 3 coenzymes. And once you're done making all those flashcards, remember to go finish all those practice problems in the study guide.